Hello everyone, welcome back to my channel. I'm starting a new series on here called Journal Club. And if you're familiar with it already, Journal Club is just a way for us to read scientific papers together and better our scientific communication and also learn how to read science papers, etc., etc. So if you're already in a research setting, you're probably already familiar with journal clubs. But the way I'm going to conduct these journal clubs is I'll have on the left side my Notion open. And there I'm going to take notes and stuff and have a list of questions I have about the paper, etc., etc. And then on the right hand side, I'll have the paper on screen. So we can read it together. And each video, I'm going to focus on one section of the paper, and then in each subsequent video, I'll go through the entire paper with you guys, just so you can get a sense of sort of what new technology is being developed in the field of biotech, medicine, biology, computer science. Those are the papers I'm going to focus mainly on in Journal Club. And yeah, so why don't we just get started right now? This video is just going to focus here on the abstract of this paper. For this week, I chose a paper from Nature Biotechnology, and it's titled scDesign3 Generates Realistic In Silico Data for Multimodal Single-Cell and Spatial Omics. So I'll write this in my notes app, in my Notion. scDesign3 generates realistic in silico data for multimodal single-cell and spatial omics. So as I said, we're going to focus on the abstract for this video. In a subsequent video, I'll focus on the introduction, and then in the next video, focus on the simulation or like the experimental design, interpretation, the data, and then the last video will just be the conclusion or any sort of biological insights we might have from this paper. So let's get started here in this video with the abstract. So the authors are presenting a statistical simulator. Oops, how do I highlight? They're presenting a statistical simulator that's called scDesign3. And scDesign3 generates realistic single-cell and spatial omics data. So the goal of this paper, the authors present a statistical simulator called scDesign3. And the simulator, it creates realistic genomics data, both at a single cell level and a spatial omics level. So the simulator includes various cell states, experimental designs, and feature modalities by learning interpretable parameters from real data. So what we have is a statistical simulator. It creates realistic data by learning from real data. And I think another important thing to note is that they're using a unified probabilistic model for single cell and spatial omics data. and it infers biologically meaningful parameters, assesses the goodness of fit of inferred cell clusters, trajectories, and spatial locations, and generates in silico negative and positive controls for benchmarking computational tools. So the simulator is a unified probabilistic model. I'm going to note, actually, some questions I have just from reading the abstract here. So I'll make a new heading here in my Notion called questions. And I like to put my questions in a toggle list. So I think the primary question I have here is, how do I make a sticky? Here we go. Oops. Why is it doing this? Okay. What is a unified probabilistic model? Another question I have is, where did it go? Sorry guys, bear with me as I try to figure out how to use this. Why is it opening up a new thing every single time? Okay, whatever. I'll just note it in my Notion. So one of the questions I have is, what is a unified probabilistic model? And I guess to make the question more specific, in the context of deep learning and machine learning, I think another question I have, I'll actually note this in pink, and I'll note this in pink as well. So another question I have is, what does benchmarking mean in the context of genomics / deep learning / machine learning? But yeah, I think that's it for Journal Club today. We focused primarily on the introduction, and we know that the authors are trying to present a statistical simulator called scDesign3. And it just generates realistic data at a single cell and a spatial omics level by learning from real data. And computationally, the simulator is a unified probabilistic model, which I have to look more into. And it helps generating cell clusters, cell trajectories, spatial locations, and it also generates negative and positive controls for benchmarking. And I don't really know what benchmarking means in the context of genomics, deep learning, or machine learning, so I'm going to look more into that as well. But that's it for this video today. In the next video, I'll focus more on the introduction, and we can continue reading the paper from there. But If you like this video, be sure to like, comment, and subscribe, and stay tuned for more journal clubs. But yeah, have a good day.